let me just see. Can you hear me now there? Am I audible from the, I'm just keeping the phone just to make sure I'm audible.
Sir, you are audible, sir.
Oh, perfect. All right. Am I visible? My video probably is not running yet.
No, sir, you are not visible, sir.
All right. I will, are we starting now? Should I go on the video or should I wait? Hear me now there? Am I audible from the, I'm just keeping the phone just to make sure I'm audible. You are audible. Oh, perfect.
A very good afternoon, one and all present today. Today is the third technical session of our webinar series, Kindling Curiosity, Ushering Young Minds into Research, organized by Department of Zoology, Rajadhani College, in collaboration with His Deemed to be University. I once again welcome one and all present today. May I now request Bandita Madam to kindly present the address of our keynote speaker today. Madam, please.
I'm delighted today to introduce the keynote speaker of today's session, Dr. Renjith Methew, a distinguished scientist and academician at NISER, Bhubaneswar. Dr. Renjith completed his MBBS from Mahatma Gandhi University, Kerala, and Ph.D. from Molecular Biophysics Unit of Indian Institute of Science, Bangalore. This was followed by his postdoctoral work at Penn State. At Penn State Hersey Medical Center, USA, and European Molecular Biology Laboratory, Germany. During his postdoctoral research, he studied cell differentiation and complex cell morphogenesis using a variety of systems, ranging from culture cell to Drosophila. His research has been disseminated through research paper published in international peer-reviewed journal. He is currently a group leader at School of Biological Sciences at NISER, Bhubaneswar, and Institute's Dean of Students Affairs. His research group addresses questions related to establishment, maintenance, and degeneration of complex cell and tissue architecture, primarily using Drosophila as a model. For those who have witnessed Dr. Renjith's deliberation, he is a highly acclaimed scientific storyteller whose stories are fabricated around scientific data and evidences. I personally vouch for the fluidity of his deliberation. We are extremely fortunate to have Dr. Renjith Methew with us today and share his thought on the topic, What's Biology? A Personal Take, a short story of a novel membrane traffic pathway. And now I hand over the platform to Dr. Renjith Methew so we may hear from him. Sir, the platform is all yours.
Thank you, Bandita ma'am, for your kind introduction, and a good afternoon to all of you. I hope I'm audible to everyone. Am I audible?
Yes, sir, you're audible.
All right. So,
Yes, sir.
Yes, sir.
And thanks to all the organizers for this opportunity to talk to you. I love interacting with young students, and I've been given to understand that this is meant mainly for young students, early graduate students, etc. It would have been lovely if I could actually have interacted with all of you, but given the circumstances, this is the best compromise we have. So as you have seen, let's get on to the talk. As you have seen, as you might have noticed from my title, there are two bits to what I'm planning to talk to you about today. The first bit is about my personal take on what biology with respect to the education and research is, a very personal vision of biology. And in the second, that's what I persuade you with in the first bit. And in the second part, what I would like to do is to give you a bit of a flavor about the kind of research that we do at our NISER laboratory. Now, usually when I have this pattern of seminars, not usually, always. This first bit goes as an interactive session where we progress with questions and answers with the audience, and the only thing that comes between the audience and me is chalk and board. But in this medium, that's not going to be possible. So I have a problem. So I just have to talk to you. Now, if I just keep talking to you, it's not going to be highly engaging, is what I'm afraid at some point of time. So then I have a problem that I don't have slides, because I never make slides for this first part. I've put together some slides. Before I go on with the slides, the first part slides are what I have put together with images kind of related to what I might be talking about, but it's not something that you need to extremely focus about. So let me just try to share my screen now. Is my screen visible? It's all good?
Yes, sir.
Yes, all right.
All good, sir.
Okay, so like I said, let's start with the first part, which is about a personal take on biology. Now, when I say that, you have to understand that I'm not a very well-trained biologist. My last formal biology training was more than 20 years ago when I did my 12. So this is, but you all know that this is the age of biology, right? Biology is changing the way we are thinking. Biology is changing the world as we know. Biology is changing each one of our lives. We know this because it's in the air and we hear it all the time. But what does it actually mean? And take 10 seconds to think about it. What does it mean when we say this? And if you were to compare yourself and your own lives with, let's say, the lives of your grandparents, or great-grandparents, what has biology done to your life that's so dramatically different? There are many things that you can think of, I'm sure. Each one of you can think of many things. But let me tell you something that's very deep and profound. At least from my point of view. I'm assuming here that most of the students, at least in this group, belong to the late teens or early 20s. Assuming that, if I were to talk to you, let's say 50, 100 years ago, even 50 years ago, I would be telling you, ladies and gentlemen, half your life is over. There's only about this much time left, so perhaps you should get serious about it. But today, I have the opportunity to say that girls and boys, what 20% of your life is over, 80% is still left. Think what you can do with it. Now, this is very deep and profound. Half of your life is over. A life expectancy of about 40 years at the time of our independence versus a life expectancy of 80 or perhaps 100 by the time you grow old. That's very dramatic and very profound. And it's not just about human beings living longer. It's about the influence that has had on all aspects of this world. Let me explain a bit more. Think about it. Think about all the stupendous achievements that humankind has made in the last 100 years, from the smaller scale of a chip to let's say, space stations. Think about all the achievements and think how much of this would have been possible if all that a woman and a man had to live was 40 years, and most of it worrying about diseases and infections. Think about our achievements, think about what we are doing to the world, think about how we are changing the world. How much of this would have been possible? And that's biological research. This is the kind of impact that has. Now, when I start a talk with this idea, I hope you're able to appreciate what I say. When I start a talk with this idea, people immediately tend to imagine that I'm going to talk about applications of biology and drug discovery and medical research and how the treatment landscape is changing. Totally untrue. Because what I'm going to talk about, or what has really, from my perspective, what really has changed the way all these affects us is the way research into biology has managed to reveal the fundamental aspects of how life operates, the mechanisms of life, the how and why of life. And this is perhaps currently best exemplified by this. The recent Nobel Prize in chemistry that tells you the impact of biology, it's actually biology. These two lady professors received the Nobel Prize in chemistry. And if you look at the declaration, it's about how their discovery leads to gene editing tools that will allow us to change genes the way we want. It really will allow you to play with genes. It's going to change the way we manage things in biology, not only biology, it's going to change the landscape of treatment of genetic disorders. But they did not discover it for that purpose. There were two scientists trying to understand how bacteria fight viruses. The mechanisms of that warfare, that's what they were trying to find out, driven by the curiosity to figure it out. And this fundamental discovery in 10 years has led to something like this. So this is what I'm trying to say. It's the curiosity-driven research into understanding the fundamentals of life that has led to the pace at which things are being changed by biology. But does it mean that biological research has always been something like this? Of course not. As a scientific enterprise, biology is rather young. As a matter of fact, there are a lot of people who still do not consider biology as pure science. A lot of my physicist friends tend to say that biology is not science. And to be honest, very often I do agree with them. It's not really science. But then as a practice, as an art of a quest to discover how life operates, biology is very ancient. It's a very old practice, perhaps started with the first time a caveman tried to describe the life around him, describe what life was. The need to describe life, the need to understand life, that's probably what drove the research into biology for a very long time. Because life is so complex, so mysterious, and so complicated, you need to first be able to describe it before you can begin to understand and explore it. So we spend a lot of time trying to describe life, what it's like. Take it, look at it, describe it, cut it open, cut it lengthwise, cut it breadthwise, describe it. Find the relationships, link it up, catalog it, make a list. This is probably what went on as biological research for a very long time. And that was important. That allowed us to understand, to basically understand what it looks like before trying to get into the details. And for the longest period of time, absorbing this knowledge, absorbing this description and the catalog is what went around as learning biology and research in biology as well. And that's not to say nobody did anything quantitative. There were, of course, just to take two random examples, what Darwin did was inspirational. I hope all of you are familiar with it, at least the basics of it. And if you look at the Mendel's experiments, what he did would stand the test even today. Highly quantitative, measurable experiments. But for most part, research for the longest period of time in biology tended to be about describing things. But somewhere along the line, it started changing. But education in biology continued, I believe, for a very long time to be about transmitting these descriptions, absorbing these descriptions. And it, to be honest, was the kind of biology that turns a lot of people off. Honestly, it turned me off, like I said, when I had formal biology back when I was doing my 12. So as this continued, somewhere along the line, biology slowly changed in the last century into something that was trying to understand the how and why of life, trying to figure out the nuts and bolts, trying to explain the mechanisms that operate behind life's processes. And it slowly picked up pace, and at some point of time, it really started running ahead into this extremely fast science that makes me outdated as I speak. Now, if you ask the question, what made this change in biology? What made biology pick up pace like this and make this the age of biology? Again, we can think of a number of reasons, but let me present what I think to be the core events behind biology picking up this monstrous pace. For that, we need to go back to what research in biology is about. What does a biologist want to find out? Explain. The biologist wants to explain a biological function. How does it happen? If you will, why does a dog wag its tail? How does it wag its tail? Traditionally, there are two approaches to finding this out. One is to take the organism and grind it, chop it up, fractionate it to the smallest component that can still carry out that function. That would be the top direction, that is the factor minus the organism. In the test tube, isolate the smallest component that can carry out the function, perhaps a protein that causes movement. Yes, that kind of research leads to that smallest component, which most of the time is a protein. And this kind of research is what we call biochemistry. There was another set of scientists who had a very contrasting way of looking at the problem. In order to identify the function or what is responsible for the function, instead of identifying the factor, they would go to the organism or an individual organism that doesn't have the function. Okay, organism minus the function and presumably minus a factor responsible for the function, which is an individual with a defect, a mutant. Analyzing mutants that lack the function in order to understand what is responsible for the function. And this kind of analysis using mutants, as you can now imagine, you all know this identification after mutation. They didn't know it was a mutation at that point of time, but that leads to identification of genes. And this is the kind of research that you call genetics. The problem is, for most of the time in the beginning, this started, let's say, beginning of 1900s. Well, Mendel started genetics without really knowing he was starting genetics. So genetics started around beginning of 1900s, similar biochemistry. And for a very long time, they didn't have a language to talk to each other. They didn't know they were working on similar things. They thought they were competing. They were competing theories, and they were bent upon proving each other wrong, then talk to each other. And that took a long time until the topic of molecular biology was founded. DNA, the genetic code, the genes are the DNA coding for proteins, and it's all related, and bingo. 1 and 1 added up to something a lot more than 2. They realized they were working on the same thing, or rather, two faces of the same coin, if you will. So everything was put together, and we had this huge wealth of information by reconciling what had been found so far and opening a new vision of how to approach problems. But still, it didn't take off. In order for biology to take off, what was required was the ability, the skills, the tools to manipulate life, to manipulate genes, manipulate DNA. That's what we call recombinant DNA technology, or what's known as biotechnology or genetic engineering. It's all about the tool set to change genes. You could change genes, you could make mutations, you could produce the protein you wanted, you could test your hypothesis. And now the field was set to explode. These probably were the core events that drove the pace of biological research to enormous rates. And from then it took off. From then you have genomics, proteomics, everything, research that goes really literally at a pace that I get outdated as I speak. Because you don't know, each second papers come out, and the amount of data that's being produced is enormous. And it is all from the days when we were simply trying to describe the structure of a flower, of a structure of the gut. It's a far cry that we are trying now to explain how things work, why things are the way it is. So that's what has changed biology. And now the point is, I kind of want to summarize this bit now. The point is, I don't really know how much of this, considering different curricula, how much of these mechanisms and how much of this hows and whys have trickled into the curriculum. My suspicion, and that's based on my interaction with the students that enter our system after 12th, my gut feeling is that a major proportion of our curriculum still is devoted towards the descriptions. And I'm not saying that descriptions are not useful or they're not important. Of course, you need to understand some other descriptions in order to imagine constructively. But at the same time, perhaps the proportion of effort that we are spending on descriptions can be reduced, because we are also fortunate to be at that point of time where we don't need to have it all in our mind absorbed. It's there at your fingertips a few clicks away. So we are now actually in a position to relegate a lot of that description and cataloging to a storehouse and spend our time and efforts in understanding the mechanisms and the experiments and focusing on explaining how things work. Now, this is really a dramatic change in priority that biology needs to make in terms of education and research. So now, I just had to see what I was going to do next. So now, with that, I kind of wrap up the first part of the first bit. What I would like to do in this first part now, the rest of the time, about 10 minutes or so, before I move on to my research, is to give you a few stories. And the objective of giving you these stories, and these are stories really from research, and the objective of giving you these stories is in order for you to appreciate how all biology actually can influence us and how dramatically it can influence us, and at which all scales it can influence us and our lives. Okay? Now, moving on to the first story. The first story is about a friend of mine. He worked with me while I was at EMBL, and currently he's working at Switzerland. So before I move on to what he's doing, let's think a little bit about a cell. You all know what a cell is. And let's think about how a cell moves stuff within it. Because a cell is like a factory. It's constantly making things, proteins, nucleic acids, and this stuff that it makes. And then, of course, higher order structures. Proteins get assembled into bigger enzymes and complexes, then there's lipids forming vesicles, and all this material get synthesized at a particular location, and then it needs to move to its correct target location in order to achieve its function. A protein needs to go to the right place in order to carry out its function, otherwise the cell is going to get screwed up. So trafficking material within the cell is a major job of the cell. And the cell achieves this through a few things. One is railway tracks. The railway tracks of the cell are called cytoskeleton. You must have heard of this. Actins, microtubules mainly. There are a few other things, but actins and microtubules mainly. And on these railway tracks are trains. And these trains are called motor proteins. These are motor proteins. Examples are myosin, kinesin. These names are not important, but just for you to have it in your mind. These are motor proteins that move on these tracks, and they have specificity. They have specificity with respect to what kind of tracks they will bind to, which direction they will move towards. They have specificity with respect to what kind of cargo they will carry. And these specificities come from protein-protein interactions. Okay, I'm not going to go into any more depths. So this whole thing is worked out. How all these interactions take place, how things move, it has been worked out. And this first movie which was running, it actually, let me just drag it back. what's moving inside? These are actually bacteria called Listeria moving inside a cell. That's what you see in the movie. But just forget it. The bacteria is using the tracks and the trains for moving. Okay, this is an example of movement within the cell. And what you see below, it's a cartoon model of the train called kinesin, which is a motor protein that moves on the track called microtubule, how it uses the energy of ATP to move. That's a mechanism. And what you see here below are the protein interactions involved in establishing these interactions that drive the movements. So we know this. We know this is one of the topics that we know really well, how these tracks are organized, how these machines are organized, and what are the factors determining their specificities, their movements, their rates. And it's also interesting, so before I go ahead, this took about 50 years of fundamental research, perhaps hundreds of thousands of PhDs spending their time working out those minute interactions between proteins and proteins to finally work out this movie, this whole thing for us. Okay. And interestingly, this particular machine here is more than 90% efficient with respect to conversion of energy to work. The best internal combustion engine that drives a motor vehicle that human beings have produced, I believe, is like 35% or close to 40% by Toyota, normal car manufacturers. But what you find on the road is probably close to 15% efficiency, 85% of the energy we waste. And kinesin runs with more than 90% efficiency of conversion of energy to work. Okay, it's just a snippet. Now, I told you this is fundamental research that has explained all this. But now coming back to the story of my friend, what he's doing. So we have this fundamental research, and what he's trying to do is to make a chip. This is not his work, and he's not the only one doing it. This is the best figure I found. There are a lot of people trying to do this, but I'm just using my friend's example. What he's trying to do is to make a chip. What this chip does is it has a spot where you can put a drop of blood. And buried in this spot are a lot of motor proteins, the trains. And this chip is full of tracks, the railway tracks, the cytoskeletal proteins, cables. And they go to specific reaction centers. The moment you put a drop of blood, it activates the motor proteins there. And they've been designed to have specificity to bind to specific components of the blood. They carry, because of the design specificity, the specific components along specific tracks to specific reaction centers. And at those reaction centers, you would get specific reactions producing an optically readable output as light or absorption. Bottom line, you put a drop of blood on this chip and stick it into a machine that measures this. It gives an output that replaces all the labs in a multi-story laboratory. One single chip renders one huge lab unemployed. Finished. Closes a complete field of employment. Of course, it opens another field of employment. It's not all negative. But you see how it's changing. It's a small thing, but how dramatically this entire research that we are standing, this fundamental research that we are standing on the shoulders of, how it's changing that field. Now, this is just an example. There are numerous fields like this, right? Now I move to another story. Now, we were talking about intracellular proteins and protein-protein interactions at the scale of microns to nanometers to at the scale of the bonds would be angstroms. That's the scale we were operating. Now let me tell you a very different scale of the influence of biology and the knowledge of biology has. This is about a virus called Renjith. It's a virus that infects ungulates. Ungulates are animals with hooves, cattle, deer, whatever. The interesting thing is this virus called Renjith has been prevalent in Asia and Europe for a very long time. So the cattle or the ungulates of these continents have co-evolved and developed resistance to the virus over millennia. But till 19th century, the African ungulates never were exposed to this virus. At the end of 19th century, 1880s, somewhere around that time, British soldiers introduced infected European or Asian ungulates to the African continent as a part of their expedition, somewhere at the top, northern Africa, I believe, Egypt. In a matter of years, Renjith decimated Africa. The African ungulates had no resistance to it. I think in a decade it crossed Sahara and was there in sub-Saharan Africa. It destroyed the cattle, domesticated cattle, destroyed the farmers who depended on the cattle for agriculture, destroyed the nomad populations of Africa who depended on ungulates for food. It destroyed the wild ungulates, deer, wildebeest, whatever you can think of. It changed the face of the continent. There were famines. Huge number of people died. Numbers are huge. And as people died, the people going into the forest, reduced people withdrew from forests. And as the ungulates died, trees started growing thicker because the wild ungulates feed on the trees, the forest grew thicker. As the forest grew thicker, tsetse flies started, the carriers of sleeping sickness, started becoming denser because the forests are their homes. And since the ungulates were gone, the carnivores, cats, lions, of course, they were still hungry and they started attacking people. There was an epidemic of man-eating lions. And people withdrew from the forest even more. You see, slowly it's happening over the years. Slowly, the ungulates kind of came back because they started developing resistance. And the lions withdrew from man to the deer because apparently deer meat tastes better than human meat. That's what the experiment shows. I don't know. But people couldn't go back to the forest anymore because they were now full of tsetse flies. Dense forests full of tsetse flies forming those death valleys of sleeping sickness. People completely withdrew. You see, a sequence of events happened, and you know what you got at the end of it? You got along the Renjith invasion, you got the famous national parks, the dense national parks of Africa. It totally restructured the biogeography of Africa, the entire continent. It stabilized recently, end of 20th century. The entire national park system, those dense forests were established due to the disturbance to the biosystem by introduction of a virus that the system was not experienced with. So just to tell you how large is the scale at which biology operates. And just to imagine, if you have this knowledge, it's great power, which can be utilized in many different ways. So that's another scale at which biology operates. And there is a lot of work that went into the mechanisms that operated behind this, the co-evolutionary mechanisms, the immunobiology mechanisms, huge amount of research. But this is the scale at which biology operated in this case. Okay. Now I'll tell you one more. I think I have time for 10. I have time to introduce one more quick story before I move on to the second part, which is our own research. Okay. Now, this story, the question that this story addresses is, are your genes connected to your behavior? It sounds like a very simple question, which you can simply answer with yes, of course. there are implications that we often do not imagine. Let's think about this story. This happens at a high security prison in the United States, and I believe sometime in late 90s. The reason that you go to a high security prison in the United States is because you commit some heinous crime. And this particular person who we are talking about, you can call him a hero if you want, but not really a hero, because he was in the prison because I believe he raped and murdered someone. And he was, and in the Supermax prison, he was awaiting his execution. He had a death sentence. And of course, it also means he had a lot of free time. That's an advantage of being in a high security prison. So with all this free time, he started thinking, okay, and he realized that his family was so messed up. He was like this, and he had an uncle who had done some heinous crimes, and then his family was full of people doing something or the other. Murder, arson. So he thought, gosh, something wrong with my family. But he was sensible enough to sit, and he started drawing a family tree. And the pedigree chart that I have put here on the screen is a random pedigree chart I got from somewhere today morning. That's not the chart he drew, but he drew a diagram of his family, marking all those people who had done violent crimes. And he also was sensible enough to go through newspapers and find out someone, I believe in one of the universities of California, was a human geneticist. So he was sensible enough to send this diagram to the scientist. And the scientist looked at it, and it looked somewhat like this. And those who know a bit of pedigree analysis would identify that this suggests X-linked recessive inheritance. He got curious. So he sent the student on a dangerous mission. So the students had to go, and whoever was still alive in his family and had done such crimes, as well as controls who had not done crimes, their blood was collected, and he analyzed their genes, their DNA. The bottom line is this. He identified that all these people had a recessive mutation on the X chromosome on a gene that coded for an enzyme called monoamine oxygenase. And the role of monoamine oxygenase is to metabolize neurotransmitters after they have been released into the synapse. Bottom line, it messes up, this mutation messes up the homeostasis of neurotransmitters, and it causes the individual to react abnormally, excessively to exciting situations, emotionally exciting situations. Clean research, very good data, fantastic publications. But there is one major problem. Is that guy guilty? Ask yourself, is that person guilty? Of course he is. Yeah, he raped, he murdered. But is that person as guilty as someone who did the same without the mutation? Ask yourself for an answer. See, I don't have an answer for this. Okay? Nobody has an answer for this. But the day is not far away when we start facing this question, but Your Honor, it's his genetics. My party is not guilty. And the society will have to answer. And no society is ready. Ours, Western, society keeps brushing it under the carpet to such a degree that it is very, very difficult, for example, in the United States, to get funding for research looking at the relationship between genes and behavior. Because we are still afraid to find this out, because this questions the fundamental tenets that organize our society. It questions what we imagine to be right and wrong, what we imagine to be just and unjust, what we imagine to be fair and unfair. It questions all those cornerstones of the society. But But I don't know for how long we can keep brushing it under the carpet, because one day it's going to get revealed. And that day, the society that has actually started sitting up and addressing these questions and wondering how to deal with it would leave behind a society that's afraid to face these questions. Okay? This is another example for you to appreciate the depth at which modern biological research is going to affect you during your lifetime. Now, we can just go on. We can just go on. I have numerous examples that I use when I take introductory biology class to my students. So I'm going to wind up here with the first part of the talk. Okay. So I hope I have another 15 minutes. Yeah. Okay. So let's just wrap it up, and then we'll go to a discussion session rather than interrupting in between. I'm going to stop that first bit of my take on biology and persuading you about how impactful modern biological research is going to be. It's like I said in the beginning, biology is not like a science, but it's unlike any other science. It doesn't matter what your persuasion is. It could be a physicist, chemist, mathematician, economist, whatever. But if you go out there and ask a question about life and how it operates, and find out a little bit about that, you become a biologist, right? Now, if you have questions about what I discussed, we'll take it at the end. I'm going to move on to a little bit of something that we do in our lab at NISER, right? Okay, so before that, I must thank for what I talked about so far, primarily to the numerous teachers that I've had over the different phases of my career, whom I cannot name. I've been highly influenced in my thinking because I never had formal biology education, and I built up my biology purely to sustain my research. And these are some of the books that have influenced the way I think on the topics that I discussed so far. Okay, now to move on to the next topic. What we do in our lab at NISER. So our interest is, you could call it cell biology, but it's quite broad. Our interest is, let's say, in understanding how cells achieve their shapes and how cells organize into tissues and how tissue shapes and structure contribute to function, and how degeneration or deformation or laws of this organization contribute to functional laws and perhaps diseases. So that's a broad outline. But I'm going to talk to you today about a small bit of that, and that's about cell shapes. Now, you see, you all must be aware that cells have very diverse shapes. That's a bunch of examples that I've put in here. There are all very diverse shapes. Now, why is that? Why do cells have all these complicated shapes? Rather than, other than just being aesthetically appealing in a picture like this? What you need to understand is that the shapes are very intricately linked to their function, and the shape has a very critical role in the function that the cell carries out. So today, what I'm going to discuss with you is about the case. It's about the case of research where we are looking at how a cell with a complicated shape does certain traits to achieve that shape, all right? And this is a cell of our interest. I'm not going to go into depth of this. We are going to go through a surface story. This is a cell which initially looks like this round cell. Here in gray, you see the cell membrane, and in red, you're seeing the nucleus. This round cell becomes this highly elongated cell in about 1 to 2 hours time. And this is a cell which is very critical for respiration. It is in the respiratory system of a model organism called Drosophila melanogaster. You might have heard of it. It's a very classical model, fruit fly. These are all immaterial. All that we need to know today is that there's this round cell which becomes highly elongated. And the complexity of the shape is not only in its elongation, it's also in what you see as this white indented line inside, which is an intracellular tube that's also bounded by plasma membrane. Okay, so that's what's represented diagrammatically here. And now to imagine topologically what it is, you just imagine a long balloon. That's a cell. And you poke your finger into the balloon, extending the plasma membrane into a long tube inside the cell. That's what the cell is like. And this long tube inside is very critical for respiration because that carries air, all right? So the cell needs to elongate and at the same time elaborate this very long plasma membrane bound tube of a very distinct shape and size inside this. Okay. Still, the last thing I want to add at this slide is that, so you have to imagine that it has two different plasma membranes, the outer and an extended inner. And the other thing is, these two plasma membranes are distinct. I won't go into the details, but for the function of the cell, the chemical differences between these two plasma membranes and maintaining it is very important. So the cell needs to elongate, elaborate this complex shape of an internal plasma membrane tube with a very distinct chemical property. So this is a very demanding job to do in 2 hours' time. How does the cell manage it? It needs to grow these two domains. When I say two domains, the outer domain and inner domain, at a very specific rate in order to achieve this specific shape in a short period of time. So here's a movie that shows the cell growing. And the signal that you're seeing is all from the plasma membrane. We are using a microscope to really watch this, label this cell, its membranes, and watch the activity of this membrane as the cell is growing in real time, which you see at the top of the movie. And what you see is a lot of vesicles, a lot of membrane vesicles moving around as the cell is elongating, and this tube of a very particular shape is growing inside it. So I'm not again going into great details, but what we have summarized here in these is that when we measure the movement of all these vesicles, what we find is that vesicles tend to have a net forward movement. As the cell grows, they tend to grow to the tip of the cell and accumulate there. Okay, when we look at it more carefully, what we see is throughout this growth, you have this tip domain of vesicles, all right? And when we look at different snapshots of the movie at different time points, we analyze it in details with using labeling this. In the previous movie, we just saw the membrane. Just imagine why now we label the cell with different cellular components to see the behavior of different cellular components and look at the properties. And what we find is that the material seems to be derived from this inner tube, popping out of the inner tube as vesicles and moving forward to accumulate at the tip as a blob of membrane. And when you zoom out, this blob is heterogeneous with different properties. Interestingly, this blob at the tip of the cell, which is independent from this inner tube, tends to also carry properties of the inner tube. Remember, the two domains have distinct properties. Certain characteristic proteins which associate only at the inner tube, we are finding at this tip blob. Okay, keep it in your mind. So now, if the inner tube, this domain, which is a plasma membrane extension, remember what I explained, if that is contributing to these vesicles that you see moving around, there's only one way plasma membrane can contribute to vesicles in the cell. That's by a process called endocytosis. That's a process by which here from the plasma membrane, a vesicle bleps into the cell and is moving inwards forget the details here. It just bleps or pops as a vesicle from the plasma membrane into the cell. That's called endocytosis. Now, what we saw here suggests that endocytosis contributes towards all these vesicles. If that's the case, now you want to find out, is the process of endocytosis contributing to all this vesicular traffic? How do we find out? We will find that out by stopping endocytosis. If you want to find out, is endocytosis doing something? Stop endocytosis and see what happens. Now, this movie, like the previous movie, this is our round cell. This is another cell. We will not talk about it today. There's this round cell, which is going to elongate. And this is all normal, all right? We have done nothing to endocytosis. The cell just elongates and forms this tube that we know. Now, in this case, at some point of time, this blinking temperature at the bottom is going to change to 32 when we have a way to stop endocytosis. And all that you're seeing is membrane, and we will watch what happens to the membrane. What happens is now we switch endocytosis off by raising temperature. We have a way of doing it. We find that the cell is messed up. Instead of forming a tube, it has this huge amount of membrane accumulating inside the cell. and the outer membrane is not really growing and elongating into the proper cell. And this, we have a trick. We can do this, and then we can bring the temperature down and restart endocytosis, and then we get the normal process back. So that clearly shows that stopping endocytosis is doing something. It's not normal anymore. What is it doing? Stopping endocytosis is flattening the growth of the outer membrane, but the inner membrane, there is huge amount of material accumulated. What does it tell you? It tells you, but the total membrane growth is the same. So what does it tell you? It tells you, okay, let's just skip this slide. This was more details to find out, just to demonstrate what we did is true by electron microscopy. But let's go back to it. What it tells us is that under normal circumstances, there is endocytosis that's plucking membrane out from that inner tube. When we block endocytosis, membrane is still being delivered, but the plucking process we stopped. So membrane is just being delivered to the inner tube, but it's not being plucked off, so it's just accumulating there at the inner tube. Along with that, the growth of the outer membrane has stopped. So which means the growth of the outer membrane depends somehow on this endocytosis plucking membrane from the inner tube. So we have a system where the primary delivery of material, you see the cell makes material somewhere. It could be ER, Golgi, etc., where protein and lipids are being made, and then it's being delivered to the final plasma membrane. So the primary delivery seems to be to the inner membrane. From there, it gets ingested, plugged by endocytosis, and it's transferred to the outer membrane. And that's required for the growth. And when we block it, the inner membrane accumulates membrane material and the outer membrane stops growing. Now that's a nice idea based on what we found. But is that true? We need to test it. This is how we work. We have some observation. Based on that, we make this model. This is probably how it works. Now we need to test this model. We do a test to see if this is correct. Now remember what I said, the inner membrane, which is shown in red here, and the outer membrane, which is depicted in blue here, have distinct properties. Now, if this model that we built is correct, that the material to the outer membrane must go through the inner membrane, then if we block endocytosis, the material that is specific for the chemical properties of the inner membrane, of the outer membrane, would now accumulate at the inner membrane. Because that also needs to go through the inner membrane, right? That's the route we are talking about. We need to test that. So we tested it by blocking endocytosis. It's a complicated slide, but the bottom line is we stained for material that's generally to be found at the outer membrane. When we block endocytosis, this receptor material accumulates at the inner membrane, suggesting that our model by this test is right. We look for different material, different categories. They all now accumulate at the inner membrane. All right. So the outer membrane is called basal membrane. So basal cargo now, when we block endocytosis, accumulates at the inner membrane. It tells us that there is this peculiar route of membrane delivery operating in this case. Now, if you remember, I started by saying that this blob at the tip appears to be derived from the inner membrane somehow. This vesicle seems to be coming out. That's how we started. And it's going towards the tip. We still don't know what that's for. I'll come to that in a moment. But if that argument is right, that hypothesis, that model, that this blob at the tip comes from this endocytosed material from the inner membrane, and again, that blob would also go down in intensity of material if you block endocytosis, because endocytosis feeds this. That's what we found. Again, no details, but when we block endocytosis, the intensity of that blob goes down. And when we restart endocytosis, all that accumulated material pops out and the blob becomes even brighter. That tells you that this blob has something to do. What I'm calling this blob, this tip domain, has something to do with this entire route of primary delivery to the inner membrane and then to the outer membrane. So we characterize this in detail, and what we found is that this tip domain is actually a compartment called late endosome, which is a component of processes that an endocytosed material, endosome, undergoes on the way to the final destination. It's an important component in membrane management. So it is a specific function. We identified that. And we also identified by inhibiting this function that this function is important for what we found. If we, instead of blocking endocytosis, we just inhibit this function, particular function of this tip domain, which is a late endosomal function, we get the same character, same phenotype, which means that late endosomal function is essential for this root. Okay? Now, I'm just going to summarize. What we found is that this very complicated cell, it rapidly develops this complicated shape, which involves elongation of the cell and growing two distinct domains, an outer and an inner domain, at specific proportions. And it employs this peculiar route of primary delivery of all material to the inner domain, from there pinching it off by endocytosis, then transferring that material to the outer membrane, which we call transcytosis, and this goes through this tip block. We test it. See, this is how experiments are built. We test it. Is it true? Then we block endocytosis. We expect certain things. We found that, and we hypothesized that this tip block, based on its characteristics of late endosome, has something to do with this. We blocked it. We found, yes, it has something to do with it. Now, to just summarize our findings, two questions are still left. What is this tip block doing? Late endosomal compartment is the compartment that breaks down the membrane and protein and sorts it this way, that way. The explanation is this. Remember, I said the inner domain and the outer domain have very specific characteristics, and this is very important for cellular function. If they get mixed up, the cell is messed up. Now, if the cell has a route where it takes material from the inner membrane and transfers to the outer membrane, you see, it will also carry inner membrane material, proteins which specifically function in the inner membrane, and if they go to the outer membrane, it will screw the cell up. So the cell needs to take it to a compartment called late endosomal compartment, where it's all degenerated, and the inner membrane material needs to go back to the inner, or at least be destroyed, before general membrane material goes to the outer. Okay, that's the role. It's a cleanup step that this tip block carries out. Now the big question, why is the cell doing this? Why does the cell run this very peculiar delivery route? It just has to deliver material to the outer, material to the inner, and the cell will grow. Why is it doing this peculiar route? The answer is this: scaling, proportionality of growth. You see, in order for the cell to achieve in 2 hours this very peculiar shape, it needs to grow the two domains at a specific correct proportion. Think about it. It's just about a delivery problem. I am making all the material, and I tend to deliver it to two distinct domains at a specific proportion. I can do a trick. I give all my material to one guy. I tell him, we have a contract. Whatever you get, this particular percentage has to be transferred to the other guy. This is a deal. This percentage is fixed. If that's the case, I don't have to worry about delivering correct proportion to two domains all the time. Whatever be my rate of growth, rate of delivery, I simply deliver everything to one party. The contract of proportions will achieve proportionality of growth automatically. And that proportionality of growth will achieve the proportionality of size of these two domains, and in turn, shape of these two domains, and in turn, the complex shape of the cell. So this peculiar route is in order to ensure that membrane delivery is always at proportions and always results in the right shape at the end of it. Okay? And now you know one can ask a question: it's just a peculiar cell in a fly. True. But what we are asking are fundamental questions. And if you start looking at complicated cell shapes, there are different domains need to grow at different rates. This is going to be a fundamental mechanism employed by many such cases. And soon after we reported this a few months ago, not soon after, around the same time, another group in the US reported exactly same mechanism in another organism, in another complicated cell. Only difference, it was opposite. The material was going from outer to inner instead of inner to outer. But same principle, same objective, achieving proportionality. So what we are looking at using the model system are fundamental principles of how processes operate, right? With that, I'm going to wrap this up. I would like to thank all the people involved. Rosalind, Soumya, and Deepshika are my PhD students. Tejas, Vineet, Salil, Adishri, Abubakar, there are a few more joining now. These are my master students. And on the other side, you have my collaborators at EMBL, where part of the work was done, and then other collaborators at different parts of the country, Mohit, Richard, Tina, etc. There are many more that I cannot list here. And at the bottom, you find all the funding agencies. I think I'd like to wrap it up now, and I'm happy to take any questions.
Thank you so much, sir. It was such an interesting deliberation. I personally enjoyed it a lot, probably for the second time. I was fortunate enough once again. A few students have a few queries, sir. We'll take a few questions.
Please, I'm happy to take questions.
So Prashant Hembram wants to know what is the definition of life and how it can be explained in one sentence.
Yeah. I'm trying to make it into one sentence. One sentence is difficult, but I'm trying to think of the simplistic definition of life, because what has been experimentally shown, see, there has been, okay, let me take a bit of time to explain this. Prashant can then derive the sentence out of what I explained, right? There has been a lot of attempt still going on to define the minimum life. If you look at the work of a scientist called Jacob Szostak, you will just YouTube Szostak videos, you will find a lot of explanations of the minimum life. So in that case, the minimum life is an information that is self-sufficient to replicate and transmit from generation to generation. If you have information and that information can replicate and that information can keep transmitting from generation to generation, that is life. And this has been achieved in the lab. And that's what I'm basing that explanation on. That you have molecules that can organize as data, and the data, without any additional information, just due to the way the data is organized, can replicate, and then it needs an encapsulation to separate from the rest of the environment. These are additional accessories. What I defined as life would still remain true. Those encapsulation is in order to protect it, and then that replicated information goes to the next generation, forming another encapsulation. That's what life is.
I hope that answers the queries of Mr. Hembram. We have one another query from Mr. Vishnu Prasad Sahu. He is asking that I did have learned about why Drosophila is used in studying genetics. Now that we used it in past and we use it now, how has it changed? I mean, we have moved to a certain degree of advancement in computation and probably have learned a lot on every aspect. So how has that changed? Or we have just stayed the same on how we used it for deriving results from experiments on these. He basically wants to know the difference regarding how the use of Drosophila has been changed.
I'm trying to. Yeah. See, I'm trying to observe the question. Drosophila has not changed, right? It's still fruit fly. It's just a model, because when you're trying to understand life, you need to work on a model. What has changed, of course, is what you're applying the model for. There are reasons why each model is chosen. There was a reason why Mendel chose pea plants. There's a reason why T.H. Morgan started working with Drosophila. And the reason is relevant to the kind of experiment that's being done. For genetics, Drosophila has certain advantages. But as technology has advanced, the applications possible with a particular model will keep expanding. We start finding new models, but also new applications for the old models. Drosophila might have been used for genetics in the beginning, but in fact, it perhaps is not used for genetics the most nowadays. A very common application of Drosophila is now for cell biology, where you want to look at things live, how it's happening in life, because it allows you to, like we did the experiments, image events in life, because with a microscope you can access, because of some degree of transparency of the tissue, you can directly visualize what's going on inside. So that's an application which Morgan obviously did not think of. The point is, many of the modern applications which have evolved due to technological advancements, these can be applied to any model system. You can extract the DNA of Drosophila and do any of the modern DNA analysis. You can use Drosophila. So it's a question of, it could be for making new findings. It could be for demonstrating one of the Morgan's genetics experiments at the resolution of molecules, right? So today we are at a stage where we can take the tool and we can choose the model that we want and apply the tool we want. Because of the evolution of advanced tools, those tight boundaries that model A is for experiment X, those definitions are lost. These are highly flexible. I hope I answered the question.
I hope the same, sir. Can we take one more question?
Please.
So it's Gunjan Pareda, and she wants to know, can we inhibit a particular endocytosis using in silico technique?
I missed the middle of the question. Can we inhibit what? Sorry. I am losing your voice, in fact.
She is asking, sir, can we inhibit a particular, madam, will you take over? Is my voice
Okay,
not
I'll
audible?
take over. Hello, am I audible, sir?
Yes, ma'am. Please go ahead.
She's asking, can we inhibit a particular endocytosis using in silico technique? It is high time now. We have been exploring animal life in experimentation.
Can we inhibit endocytosis in silico? Is that what the question is?
Yes, sir.
Well, in silico, you will be able to inhibit in silico endocytosis. And not I'm trying to again understand the question. See, by in silico, you mean something that you program on a computer.
Basically, she doesn't want to use animals. She feels like we are using animals for experiments. So without using animals, can we do it?
You cannot inhibit, you can design inhibitors in silico. That we do all the time.
Okay.
We can design inhibitors, we can design drugs, we can design things in silico, but you never know what's going to happen when you put it into life. Now, I understand where that question is from. Let me take a minute to explain this. Let me first ask a question. Then I will go to explain. Let's say somebody has a disease and let's say COVID, and I did certain programming and I designed that this compound, my computer says that this compound is going to inhibit it. And I make this compound and say, you eat it now. How many people will be willing to eat it? Because if you look at the data, the noise in biological systems is still way beyond what we can handle, despite all the advancements we have made. It's just like, the Renjith story I said, you don't know how many interactions there are. Once you go to molecular levels, the knowledge that we have is even more noisy than the Renjith story. So the in silico, how would you make a prediction in silico? You have to control variables. You can also only control the variables that you know. So we use those controlled variables that we are aware of in order to make predictions. So that we use to start as a starting point to design certain things. But we have no control over what all the actual variables are in the real system. And we are nowhere near making those predictions and understanding those without actually putting in the system. So in silico is always a starting point, but before getting a compound that you can risk putting into a, let's say, human being, we will have to do real tests. We are far, far, far away from doing anything other than that. If that were not the case, we would be churning out drugs like this, right? In silico prediction takes very little time, and I can sit here and keep making drugs. The percentage, you just have to look at the statistics of what percentage of in silico designed drugs actually come out as useful, non-dangerous drugs in the market. The percentage is incredibly low.
True, sir. I hope Gunjan might have got her answer. Siddheswari Basuri wants to know, sir, what motivated you to start this particular research?
Yeah. What motivated? Well, that's complicated because, it is something that one slowly builds as one starts doing research. So one has to define as one does research and as one starts understanding what research is, one starts looking around, looking at the kind of work going on and what is exciting, what is relevant. There are a number of factors that contribute to it. Your own interest and how relevant it is and how doable it is. So there are a number of factors. It's not easy to define.
So one last question. It's from Mr. Raman Patel, and he's asking, where do you see biology in future? Will it come in handy for the human survival on this evolving Earth?
Well, it's already very handy in survival of human beings on Earth because without biological research, like I already said, the life expectancy, I believe, was about 40 years or 41 years, even as close as 1960 WHO data. So a major part, of course, there are other things too, which improve our health, like general sanitation, but a major part of the credit for extending our longevity to this very long period goes to biological research. I don't know where it's going to saturate, how long it will extend, but it's going to continue to extend our longevity and how human beings survive on this globe.
Definitely, sir. I hope all our participants, whatever queries they had got the answers. And I'm pretty sure no one could have answered it better than you, sir.
Thank you.
Again, thank you, sir, once again from all of us. And may I now request Rashmi Madam to kindly propose the vote of thanks.
Good evening to all of you. I am privileged to offer my Good evening to all of you. I am privileged to offer my vote of thanks to the dignitaries present in the webinar. First of all, I would like to express my profound sense of gratitude to Dr. Renjith Methew, the keynote speaker for this illuminating and thought-provoking talk. Sir, the talk was very interesting, and I think it is the most interesting, informative class I have, that is cell biology class I have ever attended. The in-depth knowledge based on your research finding, you have shared must be very useful for all the participants of today's webinar. Sir, the talk was very, very interesting, and I think we need to have more and more discussion in future. I think I'm not satisfied today. I need more knowledge to grasp more knowledge from you. Personally, I'm telling, but definitely it will be very helpful for all the participants to choose their research area, and the questions which has started sprouting in their mind, in young mind. That was really very interesting question. It shows how much they are interested. It shows how much they are interested to enter into the research and to share the knowledge with you all genius scientists like you, sir. It was really very interesting, and I think it's the best evening of 2020. And since you have shared the knowledge, so much of knowledge with us, sir. Thank you, sir. Thank you so much for the talk. I owe my deep sense of gratitude
Unmute, Karan. Unmute.
Sorry for the inconvenience and the interruption. I express my profound sense of gratitude to the founder of KISS and KIT, Professor Achyut Samant, who is the driving force for all of us placed in KISS and conducting many seminars and webinars and many other events. My heartfelt thanks and gratitude. Am I audible? Yes. My heartfelt thanks and gratitude to our advisor, Professor Dr. Harikrishna Satpathy, who is constantly inspiring us to conduct seminars, webinars, exhibitions, and many kind of events. And my thanks and gratitude to Professor Sasmita Samant, VC KISS, for her inspiration, constant motivation. I express my heartfelt thanks and gratitude to Dr. Sasmita Sarangi, Principal, Rajadhani College, for collaborating with us for this webinar series. Ma'am, thank you very much. I extend my gratitude and thanks to our registered KISS Professor Dr. P.K. Rautrai, Director Academic cum Principal Dr. Sujata Acharya, our Vice Principal Sir Chitaranjan Bhoi, and Vice Principal Madam Tripura Mishra for their help and support to execute any event we do here. Particularly for this webinar, they have supported a lot. Thank you, sir and madam. And I extend my thankfulness to Dr. Priyadarshini Rautrai for moderating the session, and also the organizer of this webinar, Vandana Madam, for her effort to conduct this webinar smoothly.
It will be sacrilege if I don't offer my vote of thanks to all the participants for this webinar. It would have not been successful, but it would have not been successful if they will not participate in this webinar. And I also thankful to the scholars, teachers, researchers, and all other students, students of Rajadhani College, as well as students of KISS and other college and university students for their participation. Finally, I express my gratitude to all the people involved in this intellectual business. Thank you very much. Thank you, sir and madam. Namaskar.
Thank you, sir.
The declaration.